السلام عليكم ورحمة الله وبركاته. مختبر البكتيريولوجي for this session is about Mycobacteria. Mycobacteria is a special bacteria that need to work with it in a special lab. It's called the TB lab. It is a dangerous microorganism and need special precaution when dealing with this bacteria. It is considered a biological biosafety 3, and working with this microorganism need to work through a safety cabinet level 1, and the whole lab is supplied with a negative airflow to avoid the occurrence of any accident and aerosol. Also, the worker in the lab should be a special lab worker trained to work with this dangerous bacterium. Now, general characters of Mycobacteria. Just to remind you about this bacterium, it is an acid-fast bacteria, that is to say, cannot be stained with the usual gram stain, but need to be stained with a special stain called the acid-fast stain. And one technique of the acid-fast stain is the Zeal-Nielsen staining technique. The chemical structure, the chemical composition of the cell wall of Mycobacteria is of a gram-positive type. That is to say, the chemical composition of the cell wall of Mycobacteria don't have the outer membrane or the lipopolysaccharide which is present in the cell wall of gram-negative bacteria. So as a chemical composition, the cell wall of Mycobacteria is of gram-positive type. Cell wall of Mycobacteria is thick and made or contains more than 60% of it lipids. The cell wall of Mycobacteria is a thick cell wall, and more than 50 to 60% of it is made of lipid, mainly mycolic acid. The Mycobacteria either straight or slightly bent curved bacilli. They are strict aerobes and generally non-motile. Zeal-Nielsen stain, the principle. Zeal-Nielsen staining technique is a type of acid-fast stain used to identify acid-fast organisms, mainly the Mycobacteria. Mycobacteria contain large amount of lipid substance within their cell wall called the mycolic acid. Once the Mycobacteria stained with the carbylfoxin, the first stain, this organism can resist the acid and or ethanol-based decolorization. The Mycobacteria, once take the stain, it is difficult to be decolorized, even when we use a strong decolorizing agent in form of acid or acid plus alcohol. So, for this reason, this bacteria is called the acid-fast bacilli. The acid-fast stain, the Zeal-Nielsen staining technique, is introduced by Ehrlich in 1882, and in this staining procedure, we use the first stain is the concentrated carbylfoxin, the counter stain is the Loeffler methylene blue, and the decolorizing agent is the 20% H2SO4. In the technique of Zeal-Nielsen staining technique, we prepare a fixed smear on a slide from the specimen of the patient. We take a sputum from the patient and prepare a fixed smear on the slide. And when we starting making the Zeal-Nielsen staining technique, we apply the first stain, which is the concentrated carboxyl, and we flood the surface of the slide with this stain, and we use a benz burner or alcohol burner to heat the slide from underneath. We use a alcohol burner to heat the slide from underneath till the steam is going out the surface of the slide. But we should avoid the boiling or boiling of the stain on the surface of the slide. So once there is steam coming out of the surface of the slide, we stop heating and we repeat the heating from underneath twice again till the steam going out from the surface of the slide. We stop, then let the slide to be cool for the remaining of 8 minutes. So the application of the carboxyl is for 5 minutes. The first 2 minutes we steam, we heat the slide from underneath by using a alcohol burner for the first 2 minutes. The remaining, after we finish the 2 minutes of steaming or heating the slide from underneath, we let the slide to cool for the remaining of 5 minutes. After that, we wash the stain from the surface of the slide by using water, like we use a rubber dropper contain water to wash off all the stain of the from the surface of the slide. After that, we remove the excess water from the surface of the slide, and then we apply the decolorizing agent. Here, the decolorizing agent in the Ziel-Nielsen staining technique is the 20% H2SO4. We apply the 20% H2SO4 on the surface of the slide for 1 to 2 minutes. After that, we wash off the decolorizer by using water, and we remove the excess water from the surface of the slide. Then we apply the counter stain. Here the counter stain is the Lufler methylene blue, and we allow this stain to be act on the slide for about 1 minute. After the finishing of 1 minute, we wash the slide with the water. Again, we remove the excess water from the surface of the slide, allow the slide to air dry on a filter paper, and then we examine the slide under oil immersion looking for the acid-fast bacilli. This was about the Ziel-Nielsen staining technique. The reagent used in this technique, first stain is the carboxyl, the counter stain or the second stain is the Lufler methylene blue, and the decolorizing agent is the 20% H2SO4. It's a compound stain. There is a modification of Ziel-Nielsen staining procedure is called the Kinion Modification. Here we flood the slide with Kinion carboxyl for 5 minutes. Here the Kinion carboxyl contain phenol. This the presence of phenol in the combination of carboxyl help the stain to enter inside the cell wall, inside the rich lipid content cell wall of Mycobacteria, without need of the steaming or heating of the slide from the underneath by using alcohol burner. So in Kinion modification of Ziel-Nielsen staining procedure, we use a Kinion carboxyl. The Kinion carboxyl applied on the surface of the slide for 5 minutes without any heating, because this stain contains phenol, and the phenol helps the carboxyl to enter, to introduce inside the cell wall of Mycobacteria and stain it. After finishing of 5 minutes of application of Kinion carboxyl, we wash the slide with water. Then we apply the decolorizing agent. Here the decolorizing agent is 3% HCl in ethanol. So here again another modification. The decolorizing agent is acid alcohol in form of 3% HCl in ethanol and allowed to act for about 5 seconds. After that, We wash off the decolorizing agent with water and apply the counter stain, the methylene blue. Let act to about 3 minutes. Then wash the second stain with water, and after that allow the slide to be air dry on filtered paper and examined under the oil immersion. So this is the Kinion modification of Zil-Nielsen staining technique. Here we use Kinion carbylfoxine contain phenol instead of heating of the slide, and the decolorizing agent is acid alcohol in form of 3% HCl in ethanol as a modification. If we examine Zil-Nielsen stained slide under oil immersion, the acid-fast bacilli, the mycobacteria, appear as red, curved bacilli with a granular or beaded appearance. The acid-fast bacilli appear red, slightly curved, bent, beaded, granularly appear bacilli. The length between 1 to 2 micron to 0.1 to 0.2 with bacterium. So it is beaded, granularly red stained, curved, bent bacilli with tapered end. The bacilli appear red in sharp contrast to the blue counter-stained background. The background stained with the methylene blue appear blue, and the AFB appear red, so there is a sharp contrast between the red and the blue background. After we looking for the slide, we start counting the red, bent, slightly curved, granularly stained acid-fast bacilli in the slide. There is a scoring of AFB present in the slide to assess the heaviness of the infection of the patient by scoring the acid-fast bacilli present in the sputum specimen. So we have from this table, we look for the number of bacilli seen under the microscope and the result reported according to it. If there is no any acid-fast bacilli per 100 oil immersion field, the result report is negative. But if there is 1 to 9 AFB per 100 oil immersion field, the result is scanty and report the exact number of AFB. Like we said, if we see 4 AFB in 100 oil immersion, we report scanty 4 AFB per 100 oil immersion. If there is 10 to less than 100, 10 to 99 AFB per 100 oil immersion field, the result is a +1. But if there is 1 to 10 per each oil immersion field, the result is a +2. If there is more than 10 AFB in each oil immersion field, the scoring is a +3. This is the scoring of AFB present in the slide to assess the heaviness, to assess the load of the infection of the patient with the mycobacteria. Again, this slide is stained with Zil-Nielsen staining technique. The AFB clearly appear red, slightly curved, bent, tapered end bacilli with granularly stained bacteria. Again, this slide showing heavily loaded with AFB with the nicely red bacterium, and the background and the appear blue in color. So there is a sharp contrast between the bacterium red and the background blue. Again, this is a Zil-Nielsen stained slide showing clearly the red, bent, slightly curved, taper and granularly stained red AFB. This slide of sputum from the infected person In the background, the blue background, we see a large number of pustule, the PMNs, the polymorph neutrophils, and in the streaks of mucus in the sputum, we look for the red scattered AFB bacilli. If the smear is prepared from culture, the bacilli appear short, thick, and tend to arrange in a sort of bundle. This form of arrangement is called the chord formation. The chord growth, the serpentine arrangement, occur in virulent strain of Mycobacterium tuberculosis due to the presence of a special type of mycolic acid, the 3-halose-dimycolate, which uh, allow the bacilli to be arranged in sort of bundle. This serpentine chords lead to the formation of chord formation. Again, chord formation, clearly the slide appear in form of dhafair. This slide taken from culture of virulent strain of Mycobacterium tuberculosis. In the microscopical examination report, the stain used is Ziel-Nielsen staining technique. The color of the bacilli red in color. Staining reaction acid fast shape, curved, bent, slightly curved, bent bacilli with granular or beaded appearance. The size of the bacilli usually large, long, 1 to 2 micron in length, but usually thin, 0.1 to 0.2 micron. الارينجمنت single pairs or on group وإذا كان السلايد مأخوذ من culture راح يظهر على شكل chord formation. Microscopical identification acid fast bacilli positive. Cultivation of mycobacteria. Mycobacteria need special media for their growth. There are three types of culture media used to cultivate mycobacteria. First type of culture media is the agar-based media. Originally they called oleic acid albumin media because this media contains the oleic acid and albumin in its composition. Example of the agar-based media used for cultivation of mycobacteria is the Dubas medium and Middlebrook 7H10. This media, as I said, it contains salt, vitamins, cofactor, oleic acid, albumin, glucose, glycerol. The agar-based media used for observation of colony morphology of mycobacteria and susceptibility testing. The second type of culture media used for cultivation of mycobacteria is the egg-based media. Example of the egg-based media is the Lewiston-Jensen medium, LJ medium, and the Sternberg's medium. These media contain defined salt, glycerol, complex organic material in form of fresh egg, egg yolk, potato flour, and other ingredients in various combinations. Malachite green is included in this media to inhibit the growth of other bacteria. Sorry. Malachite green is included in this media to inhibit the growth of other bacteria. This media, the egg-based media, used for primarily isolation of mycobacteria because they need a small inoculum size. That is to say, if the acid-fast bacilli, if the mycobacteria is present even in a small number in the specimen, this type of media support the growth of this bacteria and presented as the colony on this media. So if we want to isolate the mycobacteria for first time, the best media used for the primary isolation is the egg-based media. Other culture media used for cultivation of mycobacteria is the liquid media. This media enriched with either acetic fluid or bovine albumin like the sola media, or the other example of the fluid media is the Middlebrook 7H9. These media used as a backup and for testing the biochemical test to identify the species of the isolated mycobacteria. In this slide, it contains the LJ media put into test tube. The LJ media clearly green in color due to the presence of the malachite green, the stain included in this media to inhibit the growth of other bacteria. And usually the media poured in this small uh, test tube in form of long slant to allow the long application, long inoculation of the specimen from the patient, because as we said, the mycobacteria is an citric aerobes, need a well aerated area of the media to grow, so the specimen put in this long slant part of the tube of the media to allow good oxygen in the atmosphere of the growth of the bacteria. This LJ media with a clear colony of mycobacteria on the slant surface of the LJ media. The long slant surface of the LJ media allow long surface of inoculation of the specimen, because the mycobacteria is a citric aerobes, the obligate aerobes need a well aerated area, well oxygenated area of the media to get or to gain good growth in form of colony. Growth characteristic of mycobacteria. Mycobacteria is obligate aerobes. Increase CO2 tension in the atmosphere of the incubator enhance growth of mycobacteria. An incubation temperature of mycobacteria is usually around 37 degrees centigrade, and the incubation time for up to 8 weeks. Mycobacteria generally, the mycobacteria is a slow growing microorganism and need a prolonged incubation time, and again usually around 37 degree centigrade temperature of incubation. Now, according to the growth rate on the culture media, mycobacteria can be classified into slow grower mycobacteria and rapid grower mycobacteria. The slow grower mycobacteria need more than 7 days to grow, and these slow grower differentiated according to growth on solid media, like the Mycobacterium tuberculosis need 2 to 3 weeks to grow on the solid media, and the growth characteristic of this bacterium, this mycobacterium, the Mycobacterium tuberculosis, described as eugenic growth, being rough surface, hard, tough, creamy color, easily detached from the surface of the media, and difficult to be emulsified. All these characters is described as eugenic growth of Mycobacterium tuberculosis. This slide showing the rough creamy color colony of Mycobacterium tuberculosis on solid media. The other slow grower, the Mycobacterium bovis, it needs 5 to 6 weeks to grow, and the growth described as dysgenic growth, being totally different from the growth of Mycobacterium tuberculosis, dysgenic growth, and being whitish in color growth, smooth surface, stuck on the surface of the media, and easily emulsify. Other slow grower can be differentiated according to the pigment production in relation to the light exposure into 3 groups: the photochromogene, scotochromogene, and the non-chromogene. The photochromogene forming yellow pigments after exposure to light. Example of this photochromogene is the Mycobacterium kansasi. The scotochromogene group, a yellow pigmented growth mycobacteria, will be formed in the dark, which change to red after exposure to the light, and the example of a scotochromogene is the Mycobacterium sacrofolesa. And lastly, we have the non-chromogene mycobacteria, non-pigmented slow grower mycobacteria under all condition of incubation. Example, Mycobacterium avium intracellulare complex. On the other hand, we have the rapid grower. These mycobacteria need less than 7 days to grow, and the colonies appear within a few days. Example of this rapid grower, we have the Mycobacterium fortutum and the saprophytic mycobacterium. The saprophytic mycobacterium, they are found free living in the environment and usually not associated with a human disease. Under saprophytic mycobacteria, we have Mycobacterium fili present in the water and Mycobacterium fili, Mycobacterium corduni, and Mycobacterium smegmatus. Mycobacterium fili present in plants, Mycobacterium corduni present in the water and soil, and Mycobacterium smegmatus present in the sebaceous secretion of human beings. This was a rapid, quick, practical review of Mycobacteria. أشكر حسن استماعكم وإصغائكم والسلام عليكم ورحمة الله وبركاته.